introduce to you Dr. Daniel Atwine. He works at Mbarara University of Science and Technology and many other institutions in Uganda. He's an expert in implementation research, and today he's going to explain us how to perform a clinical study in low resources settings. So welcome, Daniel.
Thank you. Thank you. Thank you so much, Camero. I'd like to appreciate you and organizers of the school for giving me an opportunity to share with you quite our experience that we have had in implementing clinical studies in a low resource setting. Thank you also for the introduction. I would want to say that I've been involved in clinical research for the last 14 years, and I've been working as an epidemiologist with the Médecins Sans Frontières, or Doctors Without Boundaries, here in Uganda, and we've implemented a number of clinical trials in tuberculosis, malaria, and also diagnostic studies. So it is from that bit of background and experience that I would want to be able to have this discussion. In the next few minutes, I'll be talking about right. I just want to check my slides. Sorry. We've divided the discussion or this lecture into these subsections. So I'll be talking about implementing clinical study in a low resource setting, and we'll break the lecture in these sections. I'll do an introduction to clinical research, and then an overview of conducting clinical research in low resource setting, trying to lay a landscape of what happens. I'll mention a bit of the contentious issues and considerations that we need to be put in place when one is implementing a clinical research in low resource settings. At least some of the learnings we've had and the challenges researchers have faced in this context, and then the conclusion. So the goal of the lecture is to highlight the landscape, clinical research, key considerations, and existing contentious issues, so as to guide strategies contextualized for implementing clinical studies in low resource settings. So I'll start with clinical research. As we talk about clinical research, it can be biomedical or behavioral research studies that involving human participants. So I think that's more the context why it's called clinical. It has to do with human participants. And clinical research has two large kingdoms. It can be experimental research or observational research. The difference here has to do with whether the investigator has assigned the exposure or he has just observed usual clinical practice. So it's an investigation when the investigator exposes the people to a certain interventional exposure, which is different from the observational studies. Yeah. So this particular algorithm can help one to quickly understand how clinical research is classified. And as I said, the starting question is, did the investigator assign exposure? If yes, then that is an experimental study. It's considered an experimental study. And if not, then that is considered an observational study. Now, looking at interventional studies, let's say you want to test a device, you want to test a drug. That means you're going to expose the participants in that study with that intervention. So it will be an experimental study. But then the next important aspect of that is to find out on a random allocation. Was there random allocation of participants within the different arms of the experiment, on the different arms of the study? So if there is random allocation, then that's what we call a randomized controlled trial. And if there is no random allocation, it is a non-randomized controlled trial. So those are very important classifications when we come to experimental studies. On the side of observational studies, the question comes in whether there is a comparison group involved in that kind of design. And if there is a comparison group, normally that will fall in the class of analytical studies, because you are trying to, in one way or another, to test an hypothesis. And so it becomes an analytical study. You're looking for cause-effect relationships. And if it's an analytical study, again, it will be important to know which direction in terms of the design, which direction are you starting from? Are you recruiting patients who are already exposed, and then you're going to follow them up prospectively to identify the outcomes of that exposure? So that becomes a cohort study. And if at all you're starting from an outcome, people who already have certain outcome and those who don't have, and then you try to look back and ascertain their exposure history to try to find out if there was some exposition that is unique to those who have the outcome. Then that's a case control study. And then if at all you're dealing with a population where both the exposure and outcome have happened and you want to assess both, it's more of the cross-sectional study. And the other part of the observational study is the descriptive studies, in which there is no comparison, because we're not trying to elicit a cause-effect relationship. So that's a bit of the algorithm and simple kind of way to understand how clinical research is designed. And then looking at the level of research evidence, if you choose whatever design, it also has an implication on the level of research evidence that it will generate. The level of evidence also influences the recommendation, when it comes to what do we recommend. Would want to have a very good assessment of the evidence to be sure that it can be trusted. And so understanding the stages of evidence is very important. And on top of the level of evidence, the first level of evidence is evidence from systematic review or meta-analysis of all relevant randomized controlled trials, and all evidence-based clinical practice guidelines based on systematic reviews of randomized clinical trials. So that is the top level of evidence. And just below that is evidence that has come from at least one well-designed randomized clinical trial, and most likely, most preferably, a large multi-center trial where you're doing the clinical trial in different sites. They can be in the same country or much better internationally. And the next below that is evidence obtained from well-designed controlled trials without randomization, which we call the quasi experiments. And below that, we have the well-designed case control and code studies. And then below, we have the systematic reviews of descriptive or qualitative studies, which is also called a meta-synthesis. And below that, we have the evidence from single descriptive or qualitative studies. And there is where you find the cross-sectional studies and all those. And then at the lowest level of evidence in clinical research is the opinion of authorities or expert, what we call expert opinions, or at times we set up expert committees to give their opinion. So that's the lowest level of evidence in clinical research. And as you design your study, you definitely think about all that. And just to give a quick overview on clinical trials. Clinical trials, as we have seen, is a very important kind of design that can help to give very trustable results if it's well designed. The definition of a clinical trial, based on the National Institute of Health and actually also the WHO definition, they don't differ much. It is any research study in which one or more human subjects are prospectively assigned to one or more interventions, which may include a placebo or other control to evaluate the effects of those interventions on health-related biomedical or behavioral outcomes. So that's really a clinical trial. There must be an administration of an intervention, and there should be a control, which may be a placebo, or it may be another alternative treatment or standard of care at that moment. And clinical trials may be referred to as interventional trials. And interventions may really include a number of things, and they're not restricted to this. They can be drugs, vaccines, if you're doing a vaccine trial, cells, other biological products like biomaterials, those who are in biomedical engineering, and surgical procedures, radiological procedures, devices, behavioral treatments, dietary choices, supplements, causes, changes in lifestyles, especially when you're doing a social intervention or behavioral intervention, and process of care changes, and preventive care, and many others, depending on what you want to evaluate in a population. Now, randomized clinical trials, just to give a bit of introduction, it approximates to what we see in basic science as a controlled experiment within the lab. So we try to do a study in a normal clinical setting, but trying to build restrictions around it to be like a controlled experiment. And the hallmark of randomized clinical trials is the assignment of participants to exposures purely by the play of chance. It's very important that as you create your groups, like the group which is going to receive an intervention and the group which is going to be at the control, that the assignment to any of those arms is by play of chance, or what we call should be random. And randomized clinical trials, because of that, they help to reduce the likelihood of bias in determination of the outcomes of any certain intervention. So when properly implemented, random allocation helps to prevent selection bias, and it also if there is an engagement of blinding, like what we do at times to blind the investigators or blind the patients, it can also help to minimize information bias. And the randomization itself eliminates all potential confounding bias, factors that could have maybe indirectly explained the outcome of the investigation. They would be eliminated. They will be balanced. Those factors will be balanced in both the intervention group and also in the control group. So that's what we mean with controlling the confounding bias. Although they are the high level really of evidence, they have drawbacks that we need to really understand. And one of them is the external validity of issues of the randomized clinical trials. By nature, based on what we've talked about, if a randomized clinical trial is properly done, it has good internal validity. And there we mean it measures what it is set out to measure, but it might not have external validity. And external validity we mean that the results may not be generalizable to a broader community. And the challenge is that we restrict a lot who participates in a randomized clinical trial. There is always a long inclusion criteria and exclusion criteria. So which clearly tells you that some characteristics, some group of patients, which we find in the real life, may never be able to be part of a randomized clinical trial, and yet the outcomes of randomized clinical trials are going to be implemented generally. So unlike the observation study, the randomized clinical trials, they include volunteers who actually have to go through that kind of selection process. And normally those participants might be different from those who may not be qualifying to be in the randomized clinical trial. They might be more healthier than those who are not involved. So that's a bit of a challenge that is in randomized clinical trials. They also can't be used in some instances, especially when you have to intentionally expose a harmful substance, to someone. So you can't be able to do that. Yet observational studies you can, because you can observe people who have been infected with a certain organism, and you can know how they present, how their system operates, but you can't do that with a clinical trial because that would be unethical. And randomized clinical trials can also be prohibitively expensive, just like the cohort studies. And they can also miss out on long-term effects of an intervention because normally they have a restricted study time. So classification of clinical trials. Normally they are classified in different stages based on the primary goals and number of participants. So I think this we may be well aware that we have the preclinical phase. At the time it's important to start with non-human subjects to try to understand the efficacy and toxicity and pharmacokinetics of a drug, or maybe even a device itself. And then phase 0 it's in healthy volunteers. Again, a small number of people, health people, and just to assess efficacy and effectiveness and safety. And phase 1, still in a fairly small number of people, but we try to look at efficacy of an intervention, its effectiveness and safety, but still in health volunteers. And then phase 2 is evaluation of efficacy, effectiveness and safety, but in patients. So we begin to deal with patients in phase 2 trial. And then phase 3 is a fairly bigger study that is going to conclude on the efficacy of that intervention. So I think you can say that if you're developing a device or a drug and you want to test it, it has to go through all these stages. And the last one is the post-marketing surveillance, which is for all subjects. So these are important, and once they're being implemented in a low-income country or resource country, each gives you its unique challenges, as we shall see. So we also have what we call a cluster randomized trials, because I'll mention a bit of them. It's important that we talk about them. These are clinical trials too, the experiments in which the social unit or the clusters of individuals rather than independent individuals are randomly allocated to intervention groups. So as you can see, in a cluster randomized trial, we don't really randomize individuals to decide whether you go in the intervention arm or in the control arm, but we randomize groups of people. So just as you can see in this picture, the individual randomization in a simple randomized clinical trial, you have a pool of patients whom you randomize, and some go to intervention group, others in a control group. While in the cluster randomization, let's say you may randomize groups, like they can be villages, they can be schools, they can be hospitals, but you randomize different hospitals, different villages or schools into intervention or control group kind of hospitals. And then all the individuals in the groups will be enrolled, or they'll be exposed to the intervention if they're in the intervention group. And all the individuals in the control will be given the control. Or they will just stay in the standard of care. So that is really the difference between cluster randomized trials and a simple randomized clinical trial. So cluster randomized trials are used mainly in the education and public health research when you're trying to test interventions at a more community level. And they are well suited for testing differences in a method or approach to patient care. When we're checking on models of care, for example, when we want to change how people receive care and we want to do interventions in that kind, the cluster randomized trial becomes very useful, as opposed to evaluating physiological effects of a specific intervention. If you want to test how a certain device reacts to the body or a drug, certainly it's better to do a simple randomized clinical trial. So, overview of conducting clinical research in low resource setting. So when it comes to implementing such studies in a low resource setting, very important considerations have come into play. And these can have, they have to do with the ethical implications, the organization of such a study, and putting into context the cultural context and diversity, the infrastructure challenges of researchers. At times you don't have the laboratory or the good systems in place in any country, and then pharmaceutical companies, sponsors, and regulatory bodies, all of them can be, they can be affected, or they are, the implementation of clinical trials in such setting is very important to all of them. And globally, these regions, low resource settings, are underrepresented in research. Yet the populations stand to gain more in research because they have a high burden of disease than the resource-rich settings. And low resource countries, they offer an attractive setting for implementing such clinical trials because you find a big number of naive population, treatment naive population, and with a higher incidence of maybe a certain disease and maybe in advanced stages. So recruitment is expected to be easier and you can get your numbers a bit very fast. Researchers also find it easier to implement research in these settings because of reduction of costs in recruiting patients. And also there are weak health systems. And when you have a weak health system where it can give you both sides, advantages and disadvantages, but the advantages would be that if you are testing another mode of care or way of treating patients, it is very easy to have a good comparison and to really be able to demonstrate the benefit of that intervention. And looking at all this, it tells us that, you know, in as much as there are potentials, but there's also vulnerabilities, which any researcher needs to balance when they're implementing a clinical study in these settings. Some of the difficulties in implementing the clinical trials in these settings, they rise from problems that are related with recruitment, being able to get your participants, then obtaining valid informed consent, and I'll be talking about these, and then ethical compensation mechanisms for extremely poor populations. So you want to balance, you know, supporting a patient, but without coercing them. Then retention, being able to keep people in the trial or in the study until the follow-up time is over. Then poor health infrastructure and considerable socioeconomic and cultural divides. So you can face a bit of resistance in some places if you don't handle the cultural context well. So whether trials are investigator-initiated or driven by a pharmaceutical industry, these difficulties are quite cross-cutting. I want to just give in this slide, it's a bit a busy slide, but some examples of difficulties in clinical research in these settings. The first one is the absence of harmonized regulatory requirements and regulatory reliance. So what we find in many places that the regulatory framework is still growing and it's still young. So it is very possible that in a place you want to implement a study, there is no active research ethics committee, or it is lacking capacity to be able to review such a complex project. And then another issue can be an administrative bottlenecks, especially if you have to work within a public institution and there is no clear research administrative infrastructure. So you can struggle to implement the study because of system challenges. And then financial bottlenecks, especially when research budgets are drawn without extensive groundwork. So it happens some investigators from the north, and you draw a plan, but without knowing the context within the setting where the study is going to be implemented. So you can find that there was a lot of under budgeting because of some risks which were not foreseen. And then multiple languages and diverse cultures. This is important in the conception stage of the research. It has to do with how you develop the tools, will people understand them, are they in the right language, which language are you going to translate in, all that scale. And then compensation mechanism for research participation. This can vary across countries. Like in Uganda, we have to compensate for time, the time a person spends in coming for the visits related to your study, and also time transport refund. So all those mechanisms, and they can vary across countries. And interviewer compensation modalities also, thinking about salary scales, how you're going to pay the research team. Again, that is always an issue because you have to try to balance with the salary scales within the country to try to see how to fit in an attractive area, in an attractive amount. The need for additional documents required to ease the data collection. Most of the times there is much more you have to develop as site documents that are very contextualized in different settings and countries just to facilitate data collection. And inadequate interviewer competence. In some places, it can be really quite difficult to get competent people who can implement research well. So, which goes in for conscious budgeting, because you may have to invest a lot in training and retraining, and also capacity building capacity and making sure it is it's available in the setting you're working in. So that always goes hand in hand. And then different ages or legal consent between countries. Basically, who can consent for themselves? You know, you find in Uganda it is 18 years and above, and then in other country they may say 20 years, others 15. So when you're doing a study in a different countries, you may have to think about all that. Then not dedicated research administrators in many settings, that's also another. So you find that as a researcher, you have to also do research administration work really a lot and be involved in finance management, in controlling the budget. Yes, it's your role as according to good clinical practice, but it can give a lot, it can take a bit of your time. And there are some of the recommendations that the different researchers have really made to try to deal with those challenges. And one of them is training and capacity building for regulatory authorities so as to harmonize research regulation. So training, then independent support networks for clinical trials, working within different groups and get a bit of support through those collaboration. You can have monitors, internal monitors coming from another institution to come and support a clinical trial in another, or channeling funds directly to the point of expenditure, or developing an efficient financial management system in a country where you're doing your research, just to make sure that that process is not tedious to you as an investigator. And inclusion of local investigators at conception. This is very important, that you have a local investigator in that country, and they can guide the contextualization of the study. And financial audits, very important for accountability, and then testing and verification of translation for accuracy. So translations, and you have to do back translations just to be sure that when they have translated, the message has not changed. And testing and verification, yeah. And then preparedness and adaptability are essential ingredients. For example, you can come and do implement a study, but unexpected events can happen, like how we experienced in COVID pandemic, and things can change, and you have to really adjust a lot of things on how you implement the study. So that flexibility and adaptability is important. Then establishing research administration infrastructure, that could be more of something that needs to be pushed for and can come in as capacity building, but also, yeah. The number 9, multi-site trials should have national coordinators selected before onset of the trial. And again, they should participate in its conception. Investigator-initiated pragmatic trials are very important, especially when you do trials that are needs-based and people feel that it is really helping them and addresses the problem they have. Such studies are appreciated. And comprehensive community engagement strategies. In low resource settings, buying in with the community is very important in the success of any project. And so community engagement is key. And trials to target the specific medical needs of the country as perceived by the country. At times, alignment with the national goals or research goals is really very important. It gives the project more acceptability. And recognition of a profession of the clinical trial scientist. Yeah, these are some of the things that basically making sure that there is more capacity built in these settings with regard to people who can run clinical trials. So these are some of the recommendations that different researchers have given. Now, I will briefly jump through these contentious issues and considerations in implementation of clinical research in low resource settings, just in the case of time. So some examples, like there are still challenges in selection of study control groups in clinical trials in the context of outbreak. These are still very challenging things. Like if you're going to test a drug in the context of Ebola outbreak, like how it was in West Africa, we would know that yes, an intervention, treatment is needed to treat this disease, but there is no alternative treatment. So you find a dilemma that you have a group which has Ebola and you want to give them an intervention, but the control group should be another person who has Ebola but does not receive that intervention. But that in its case will be ethically not right. So some of the challenges that researchers are struggling with is how to choose the best control group in such a context. The next one is general participant recruitment issues. As you start a study in this setting, you want to think about recruitment and retention. Can you get your numbers? Can you keep them until the end? Can you minimize lost follow-up? And talking about recruitment, strategies have to be set from the start on how you can maximize participant recruitment into the study. And these strategies can vary according to the study population you're dealing with, the setting of the study, where it is, and the disease you are studying. Now, some of the few recommendations I can mention is that, some people try already in the design to include both retrospective and prospective methods, especially when they know that they may not have the good numbers if they're going to only recruit prospectively. So they could also make use of the records, the historical patients, especially if it's a disease, like in case of chronic diseases, where the historical patients are still patients. Yeah, so that's one way. And then trial budgets also, ensuring that, they budget and put a bit of some space in the budget, some funds that can help in pre-trial training of researchers to make sure that they really can maximize screening and enrollment of patients. But at time also what we do is increasing the number of catchment sites where you get patients or participants from. And if you're at a regional hospital, you might engage some lower health facilities and create a system of referral of patients to the study site. And that also has been helpful in our experience. And also thinking about including use of phones and web-based electronic tools where applicable. They're very useful in recruitment, but also retention. In the trials I've been able to implement about 5 trials. We use a bit of the phone, like when we have low health facility contact persons, they can call us that there is a patient, and then we can go and pick that person and bring them to our site and enroll them. So that really helps. And it also helps in follow-up. We saw that during the COVID lockdowns, very helpful. And preliminary community sensitization, engaging the community early in the conceptualization of the project is key. And they can support you, and they can also, it can increase the number of participants who come to that study. There are still issues of willingness to participate in clinical trials. And I know it's much worse with these emergent conditions like COVID-19. For example, when you're testing the drugs or vaccines. But what we have seen that even it can vary across gender. You find that males can easily accept to be part of a study based on the social benefits of that study, like money compensations but things like personal risks, when people perceive that that study may risk their lives, or you are removing a lot of blood, or you are putting a device in them and they suspect it, that can make them to not want to be part of the study. and also potential participants, if they perceive that this study is beneficial to them, it can be effective and useful, they can easily be motivated to be part of the study. So, and this is why the researcher has to really engage the community and help them understand that this is the community representatives to understand the effectiveness of the study and the intervention you are evaluating. So there are still contentious issues around the enrollment and follow-up of pregnant women in clinical trial. So there's been quite a bit of barriers and hesitancy in engaging pregnant women and lactating mothers in trials because of the fear of, their risks and also the risk to the fetus. So, but the challenge is that the pregnant women continue to receive, off-label treatments, off-label medications. Yet the data was collected from a completely different population, healthy and at times male individuals. So this has been a challenge, and I think it's not only for low-income countries, but it's a bit more broader. But we are seeing that there is still need to generate data in this group or in this population so as to improve their care. But today, what's really encouraging is that there's been a number of published guidance to researchers and industry on how to recruit and how to retain and how to monitor these pregnant women in any study, and so that data can be collected in that regard. So if you're doing a study in that area, it is quite a lot of uniqueness to think about. At times, there's a lack of harmonization. With regard to the regulation of basically how to get consent, of like children from minor parents. Minor parents is that by themselves, they are young, let's say they are below 15 years. And in many countries in sub-Saharan Africa, we know that actually there are many underage mothers, people who deliver early. And now when that mother has a baby, the regulation is a bit challenged because they are not supposed to consent themselves. Actually, they are supposed to give an assent, and there should be another person to consent, an adult who is considered by law to be an adult. So now how can a minor or a minor parent consent for a baby? So there is still a bit of challenges in how to deal with that scenario, especially when you're taking that age of mothers to be part of your study. And the guidance, really now, some of the approaches that are existing, but not implemented in every country, but they can vary in every country, that some countries consider them as emancipated minors, and they can still accept an individual consent from them. And then at times they can base consent where there is no regulation. You can base consent on the guidance of the national laws on medical care. Then in some places where there are no laws or guidance that is inexistent, you may have to get the interpretation of the local decision-making culture, like how, by engaging the community and finding out whether such a person in a community can be considered to make a valid decision to be part of a study. And also implementation of informed consent for children with minor pain may be actually variable. It can vary and hampered by absent or ambiguous clinical trial regulations in different countries. So if you're implementing a study in different settings and targeting this group, it could be quite a challenging one. I'll quickly talk about participant loss to follow up and retention as another key important aspect in this setting. people migrate, people travel, people change home addresses, and when you lose your numbers and fail to maintain your sample size, a clinical trial or a clinical research may lose its power for you to be able to conclude on the hypothesis you had and to answer your objectives. So it's an important aspect that the researcher thinks about in these settings. So some of the reasons for why people don't complete follow-up or they get lost to follow-up, at times it is the consent withdrawal. Some participants will withdraw their consent. And this is common in children trials, especially when the father was not engaged in the consenting and the mother consented for a child. So the father can withdraw the consent. And so engagement of both is important when it comes to that population. Participants also thinking that they don't need to come frequently to your research site. They can by themselves stop, and then you lose them. Others is time constraints because they are working in different places. Others migrating for better employment and travel, and also time when the travel distance to the trial site is so long. People may get lost. So some of the things we have practiced in ensuring retention is involving fathers at consenting. That's also another aspect for children. And then continuous community engagement. So at times there is a bad news or bad communication going on in the community about the study. And if you don't pick it and address it, the study can suffer. People can drop out. And then restriction in eligibility criteria. At times you know that if you engage refugees or prisoners in a study, they are not a stable population. They can leave and you can lose a number of people in your study. So also at times we use treatment supporters. If it's a treatment that we are giving someone, we ask them to identify someone in their family whom they come with. So just in case they are lost, we have someone else to call and they can help to find that patient. And adequate explanation, explaining the trial procedures well and the risks and benefits so people understand them, they don't get confused on the way. Then innovative tracing of participants. So at times you have to track them, you have to take their home detail addresses and find them, look for them. So at times that means more budget for tracing. And then good participant and research team relationship. If you don't give a good relationship between the research team and the participants, some participants can still refuse to come back. And continuous counseling and support to patients is key. And logistical support to patients is also very important. And limited radius of patients where they come from in terms of catchment area. The closer to the catchment area to your study site, the more likely you will have people stay in the study. And then phone calls, as I said, very useful. And then doing home-based follow-up is another very wonderful way for retention, and then wide stakeholder engagement and develop a participant retention plan. Yeah, I'm a bit concerned about time, so I could maybe skip some slides. So some shortfalls, some other challenges we have is if you are investigating a drug or a device, you want to detect adverse events. Normally,
another 10 minutes or so.
Okay. Now, on from our practice that how would you list those adverse events? It can be through physical. Most of the time we rely on asking participants what they have experienced. And that questioning method can cause variation in how different researchers report and how many adverse events they report. So we found that questioning the participant using a checklist, like a checklist based when you have listed each expected kind of adverse event, helps you to elicit more adverse events than if you openly ask them whether they got any problem. So that's an important thing. Other challenges in conducting clinical trials in children, I think we mentioned much of them on the other side. But when you're dealing with children in low resource settings, there's a challenge of diagnosis. Sometimes the facilities there diagnose conditions in management of sick children. You need a good hospital nearby, which you're sure they can manage them. And then large catchment areas, when they're coming from far, retention can be a problem. Adverse event attribution, at times children are prone to have many other infectious conditions when they are still in your study. At times you're not sure whether it's intervention, it is these other conditions. And there's a lot of self-medical administration in low resource settings. So again, it can confound or becomes difficult to attribute outcomes in some places. So at time what we do is that if a child person study in a trial, whatever sickness they get, they should come to the trial site, and we are the ones to at time manage them or refer them and follow up on what care they receive. So
Daniel,
all these things,
can I interrupt you?
Formulation and detection of treatment response at times is very difficult. Response in children and intervention vary according to age. So
Daniel, can you hear me?
And there's also community involvement. Community engagement, very important, as I say, in studies done in this setting, help prevent public mistrust, and it increases acceptance, adoption, and also participation in the trial. But even when you are finished doing your trial, if it's to be adopted, it's important that you engage the community well. And the major goals in this is you have to be very transparent, meaningful, and collaborative, and people should know that they are benefiting in that relationship that you're creating with them, and it's not exploitative. So another goal for community engagement is to ensure that research is responsive to the needs, priorities, and what the community expects. So there has to be a bit of alignment of expectation, and also to ensure the relevance of the proposed research to that community.
Daniel, can you hear us?
Increase acceptance, and also building, strengthening, and sustaining trust between researchers and communities. Community advisory boards are a growing thing in low resource settings now. Doing intervention must have functional community advisory, and it's a requirement by the regulatory bodies now in many settings. And so this is really important, and this group of people is coming from key people whom you would say they are representing the community. They can help in the contextualizing the documents, like informed consent, and also guiding in the conduct of the research in a more ethical way. And they can also come in handy in educating the local communities because people feel they are closer to them. So, but the setup of these committees or boards is still, in its infancy, and they suffer a lot of challenge, including the regulation that monitors them. So, other challenges that you can face in implementing studies is data integrity, ensuring that data is good. Possible, but there has to be more training.
Daniel, can you hear us?
Yeah, there have to be more training, and at times the software you are implementing in the study So this is an area that we have to talk about. So I want to, another challenge is minimizing patient ineligibility trials. So it is very common that you may screen many patients and getting verified by eligible. And in our experience, at times it causes the biggest cause is using the non-indigenous laboratory reference. If you are not choosing indigenous references, it becomes very difficult. Yeah. So I would like to maybe yeah. Because of time, I just see my time is far gone. And
Riproviamo. Daniel?
yeah, Camero, it's
Daniel, can you hear me?
The remaining part.
Daniel? Hello?
Yes, I can hear you now.
Oh, finally.
Yes.
Okay, yeah. No, I wanted to tell you to wrap up because, you know, you've gone way past your time. I kept trying to stop you. So if you can just conclude in a couple of minutes, maybe we can see if there are any questions, but we really need to go on. So can you just wrap up quickly?
Yes. Many slides. So regulatory, another challenge we face is the regulatory framework for biomedical device research in Africa. I could say that there's still a challenge in accessing medical devices and in vitro diagnostics, which necessitates more innovation and research. But at the moment, there is more of regulatory framework for pharmaceutical products, which is well advanced, but not the pathway of other health products, including medical devices, has been given less attention in Africa. So many countries, we don't have a research framework that guides, a regulation that guides research in medical devices. So if you're implementing in different studies, in different countries, it becomes very important to acquaint yourself with what procedure is available in that context. So in conclusion, low resource settings offer a good environment for clinical research, given the high burden of disease, but measures to mitigate the anticipated challenges in research implementation need to be put in place, and they can differ across countries. And we definitely need to move towards fast harmonization in research conduct and monitoring amidst this changing landscape in these settings. And also, knowing these challenges we've discussed, it is important in supporting adequate planning of clinical research in these settings. I thank you. And sorry for the time.